Hello, everybody. I'm Marcia Higas, and it's my pleasure to introduce today's seminar speaker, Raul Mostislavsky. And thank you all for joining today's Dana Farber Harvard Cancer Center Connect Science Series. Please feel free to join in on this website, and also please feel free to ask any questions via the chat box. So a little bit of background about Raul. Raul is currently a professor of medicine and scientific co-director of the MGH Cancer Center. He has a long track record of successful, really cutting-edge scientific discoveries in the fields of chromatin biology, immunology, and metabolism. I want to highlight a few things. So Raul received his MD from Argentina, and then his PhD from the Hebrew University. For his doctoral work, Raul was awarded the Amersham Science Prize, and he performed postdoctoral work at the Children's Hospital at Harvard Medical School, where he performed cutting-edge research on SIRT6 sirtuin discoveries and linking SIRT6 to premature aging and regulation of genome stability and chromatin biology. And it's a real pleasure to have Raul here today. Thank you.
Thank you. Thank you very much, Marcia. Thanks the organizers for inviting me to present my work in Connect Science. It's really a pleasure to be here. So I have a monitor in front of me, so you will see me presenting and watching to the sideways, but that's because I am presenting through the monitor. Don't feel that it's just I'm ignoring the side part of the presentation. So as Marcia mentioned, my laboratory has been working for many years on this connection between chromatin biology and metabolism. I really started a long ago through our interest in chromatin biology. And I don't know how much I need to introduce this to this audience, but you probably all know that our DNA is compacted within our nuclei in this structure called chromatin, which for many years people believe was a rigid scaffold with the only function to compact and organize the DNA within our cells. We now know that actually chromatin dynamics and the ability of the cells to open and close chromatin have critical functions in many different functions in the cell, including DNA replication, transcription, and DNA repair. My laboratory has been interested on all of these roles for chromatin for many years. And as Marcia mentioned, during our postdoc, in my postdoc, I started my interest in one group of proteins called sirtuins. For those of you that are not familiar with sirtuins, they were originally described in yeast as in a screening for silencers, actually. Many of you know about the work of sirtuins in aging, but actually these proteins are chromatin, at least SIR2, the main factor in yeast, and the family member in yeast SIR2 is an histone deacetylase, and it came out of a screening for silencers. So if you mutate SIR2, you cannot inactivate genes, and you have changes in gene transcription, changes in gene recombination in yeast. When I started working on sirtuins, we knew very little about the roles for the mammalian ones. One other feature of sirtuins is that all of them use NAD as a cofactor. So very early on in the field, people realized that these sirtuins, by working on chromatin and depending on NAD, they may be linking sensing of metabolic changes to a response on chromatin. And when we started working on them, I decided to focus on SIR6. My background was on chromatin. And from the 7 mammalian homologs, we early on learned that actually SIR6 is the main one that function primarily on chromatin. There are several sirtuins in mitochondria, and Marcia is an expert on several of those. My laboratory has been focused on SIR6, and we've been using it as a nodal point to understand chromatin adaptation to potential chromatin adaptation to metabolic changes. When we started this work, I was, again, very much focused on chromatin biology. I didn't know that the work will take me towards a path in metabolism, but I will tell you how we ended up working on that. So as I said, CIR6 is a chromatin deacetylase, and work from several laboratories, including that of Catherine Chua and Dave Lombard, show very early on that CIR6 is a very robust histone deacetylase. And so hours after you knock down CIR6, you see this major increase in K56 and K9 acetylation. These are the two main lysines in the histone H3 that CIR6 regulates. And when we started our work many years ago, we knew a lot about K9 as a major repressive mark. We knew much less about K56 in mammalian cells. And I can tell you today, many years after, that we know that both marks appear to play critical roles in gene transcription, and CIR6, by regulating those two marks, is a critical modulator of activation repression in multiple genes, as I will show you in the coming slides. So one thing we did early on is to try to understand the role of CIR6. We generated knockout animals. And interestingly enough, these mice that were deficient for CIR6 were born at normal Mendelian ratio. We didn't see major developmental defects, at least in this particular strain. And we published later other papers showing that in specific strains, CIR6 have roles in development. And I won't show you today, but actually there is a recent we described recently a human syndrome with a mutation of CIR6 that causes perinatal lethality. But this is not something I will discuss today. These animals in this particular strain are born normally, but very rapidly degenerate. And in less than a month, every one of the homozygous knockout mice die in around 24, 25 days. And the reason they die is because they develop this profound and fatal hypoglycemia. So when you remove CIR6, animals start losing glucose. Here is glucose in blood, and you can see glucose going down precipitously. And by day 24, every single one of these animals, this is 100% penetrance, the animals are dead from a coma hypoglycemia. So when I was starting my lab, I decided the first thing to try to understand why lack of a chromatin factor ends up with this unique metabolic abnormality that was so severe. And to make a long story short, because we published this long time ago, what we found is that CIR6 acts as a co-repressor of a factor called HIF1 alpha. This is a known factor in the hypoxia field. What many of you may not know, that HIF1 alpha also regulates changes in nutrients. And it's stabilized not only when you have low oxygen, but also when you have low nutrients. And in normal conditions, when you have normal nutrients and normal oxygen, CIR6 co-repress HIF1 alpha to repress the expression of glycolytic genes. And by doing so, and among them, you have pyruvate dehydrogenase kinase, which inhibit the entrance of pyruvate to the mitochondria, lactate dehydrogenase that push pyruvate conversion into lactate, and the glucose transporter GLUT1. All of these, by being repressed, allow pyruvate to go to the mitochondria for normal respiration, to produce ATP in the mitochondria. This is what cells do under normal conditions. Under conditions of stress, meaning low oxygen, so you cannot sustain the TCA cycle, or low nutrients, the cells respond by doing what is called a glycolytic switch. CIR6 is kicked out of chromatin, and we prove that by using chromatin immunoprecipitation experiments. You get HIF1 alpha to be transactivated and stabilized. These recruit transactivating factors like the histoacetyltransferase P300. You get acetylation in these genes, activation of these genes here, and a switch towards production of lactate, which is much less efficient in the ability of the cells to produce ATP, but it gives the cells an opportunity to survive under conditions of nutrient stress. So we proved this in an original paper that we published, and we explained the phenotype of the animals. The animals were dying from the hypoglycemia because several tissues, when we were knocking out CIR6, they were constitutively doing this glycolytic switch. They were uptaking glucose because of the upregulation of GLUT1 in the brown adipose tissue, in the skeletal muscle, and in this uncontrolled uptake of glucose was causing the hypoglycemia that derived in a coma hypoglycemia. The brain didn't have enough glucose to sustain life. That's why the animals were dying. I will give you a little detour before going into this metabolic feature. But just to say that when I made this diagram, which was long ago, I put CIR6 with HIF1 alpha working at the promoter. Okay, this is already outdated. I should be changing this, but I am not very computer savvy. Took me a lot to make this animation. But we published last year, when we did the first genome-wide chromatin immunoprecipitation studies to look where CIR6 in the whole genome was sitting. We got a surprise. We did not see CIR6 sitting at the promoter, but rather intragenically. And the results are here. So these are human ESers, and this is what is called a chromatin IP chipsec. Okay, you look at the whole genome where these different factors are sitting. And you can see here is a HDAC1. This is a classical histone deacetylase, which we know sits mostly at the promoter. And what you are looking here is each lane is a gene, and you have in each one of these graphs a 3 kb upstream and downstream of the transcriptional starting site. And you can see HDAC1 shows a very robust signal exactly at the TSS, which you will expect for a factor sitting at the promoter. When you look at CIR6, we see a completely different picture. See much less CIR6 at the promoter, but much more CIR6 intragenically with other chromatin factors. And more importantly, and this is from mouse ESers now, here is CIR6, the same picture with a lot of genes showing intragenic binding. This is a POL2. We see CIR6 sitting intragenically with the polymerase. So we ask, maybe CIR6 is not acting at the promoter as other HDACs in charge of compacting chromatin and inhibiting a transcriptional initiation, but rather CIR6 may be acting at a step downstream of that step and controlling what is called transcriptional pausing and elongation. And that's actually the case. We published this paper last year, so I will give you just a couple of examples. This is called the pausing index ratio, or polymerase traveling ratio. What you measure genome-wide is how much of the polymerase is at the TSS, how much of the polymerase is intragenically. And you can see here in one example that in the knockout cells, you see much more of the polymerase intragenic compared to the wild type cells. And by comparing the amount of polymerase in the promoter or intragenically, you can derive what is called a pausing index. Okay, how many of the genes have more polymerase here or intragenic? And the more you move to the left, the less pausing you have. And you can clearly see that knockout cells and wild type cells that has been exposed to metabolic stress by removing glucose, they exhibit a shift to the left, indicating that these cells are elongating much more. They have much less polymerase paused. This is a checkpoint that the polymerase, many genes, they perform, they move 50, 100 base pairs, and they stop to check whether conditions are appropriate for full elongation. We found that CIR6 as an histone deacetylase appears to control this step by retaining the polymerase in a pause mode. And if you remove CIR6, then many of these genes start elongating. And when you look at the mark that CIR6 control, here is K9 and here is K56. This is a linear plot. This is in the whole genome. Most of the genes don't experience much changes or K9 of K56 acetylation at the promoter. The main changes are intragenically. Again, indicating that CIR6 appear to control these chromatin marks intragenically. And in data, I don't have time to show you, I will just summarize these results. We found that what CIR6 does is retain what are called elongation inhibitory factors. CIR6 is involved in coming down with the polymerase and retaining these factors, among them NELF-E and DSIF. And these are factors that when they are together with the polymerase cannot move and elongate. And when the cells remove CIR6 from chromatin, you kicked out by activating a kinase called CDK9. You get phosphorylation of POL2, phosphorylation of NELF. NELF is kicked out, and you can bring now elongation factors like bromodomain 4, CMYC, PAF1, and these activate the polymerase to allow elongation. So our results describe for the first time a chromatin deacetylase. This is the first histone deacetylase described with roles in transcriptional elongation. And we believe this mechanism provides an efficient on and off switch to modulate nutrient availability and the ability of the cells to respond on chromatin by activating specific metabolic genes. So back to the metabolic feature, I mentioned that when we remove CIR6 from cells, the cells were performing what is called glycolysis, even under normal oxygen conditions. When we remove the chromatin factor, they were constantly, in a constitutive manner, making lactate. They didn't care about the oxygen levels or the nutrient conditions. Because we removed the repressors, the cells were constitutively doing what is called aerobic glycolysis. Okay, the ability to generate lactate from pyruvate even under normal oxygen conditions. We proved this in normal cells, but this phenotype of the cells, the ability of the cells to perform glycolysis under normal oxygen conditions, that is called aerobic glycolysis, has been described by Otto Warburg many, many years ago in the context of cancer. He showed in the 1920s that transformed cancer cells, even if they have plenty of oxygen, they prefer to generate lactate from pyruvate. He couldn't explain it why. He at that time thought that they have mutated mitochondria. So he said they have no choice but to perform what we call the Warburg effect, aerobic glycolysis, because their mitochondria are dysfunctional. We know now, more than 80 years after, that this is wrong. Cancer cells have perfectly functioning mitochondria, but they do this adaptation to send glucose sideways for biomass production. And many laboratories have shown why cancer cells do Warburg, and we know that a lot of this has to do with these rapidly proliferating cells sending glucose sideways to the pentose phosphate pathway to produce nucleotides for DNA and RNA synthesis, acetyl-CoA sideways for lipid biosynthesis for membranes, and even sending sideways glucose for serine biosynthetic pathway to obtain amino acids for making bricks, for making biomass. So that was the reason why cancer cells were believed to make the Warburg. Very few people studied the how. And when we had our results with the CIR6 deficient cells, we asked, maybe cancer cells get rid of this chromatin factor to acquire a metabolic adaptation. In other words, maybe CIR6 evolved to be a tumor suppressor by protecting these cells from acquiring what is called the Warburg effect. And at that time, when we started this experiment, this was a novel concept because most of the people believed that when cancer cells were dividing, they randomly, some of them acquire the ability to do Warburg, and these were pushed because they were acquiring an adaptation that was an advantage. So this was retained by the cells, and they were taking over the population. Our results suggested that this was a regulated process, and we had here a chromatin factor that may be playing a role in regulating this metabolic switch. So we started to explore this in models of cancer. As I told you, the full knockout died too early, in less than a month, so we could not use the constitutive knockout of CIR6 for this experiment. So we generated a conditional deletion of CIR6 in the intestine, and the reason why we chose the intestine was because in the TCGA datasets, from the human TCGA, we saw that in colon cancer, many of these tumors downregulated CIR6. So we decided it would be a good model to test. We made a conditional deletion of CIR6. We crossed it to the APC mean mouse model of colon cancer. Actually, the APC mean in mice give you only adenomas, not fully transformed disease. And interestingly enough, when we made the animals that carry the APC mean deletion and they were deleted for CIR6, these animals developed much more tumors, bigger tumors, and here you have an image of a full transformed adenocarcinoma. So by removing this chromatin factor, we were making the cells even to go from a non-transformed adenoma to a fully transformed one. And this phenotype was driven by this glycolytic switch. When we inhibited glycolysis, and we did that in the animals by giving these animals a drug called dichloroacetate, DCA. This is an inhibitor of PDK1. I mentioned this enzyme This is one of the enzymes that is upregulated when you don't have CIR6, and it inhibits the entrance of pyruvate to the mitochondria. When you inhibit PDK1 with dichloroacetate in drinking water, and look at the right side of this panel, these are the CIR6 conditional animals. Most of the tumors were basically cured. Most of the low grade are gone. All of the high grade tumors are completely gone. So these tumors are much more aggressive, and they are much more aggressive because in the absence of CIR6, they are exhibiting a push to be more glycolytic. Again, we published this many years ago, but we discovered one of the first chromatin factors that have the ability by itself to transform immortalized cells. You still need to get, at least in this system, you still need to get rid of the checkpoints by mutating either P53 or P16. But once you have this first hit, our tumor did not acquire second hit mutations like amplifications or CMYC or KRAS mutations. Those tumors did not exhibit those. By getting rid of this chromatin factor, we push both a metabolic adaptation, and in results I didn't have time to show you, CIR6 also co-repress MYC. So we see endogenous MYC being stabilized and ribosomal protein genes upregulated. So you get a boost in protein synthesis, a glycolytic switch towards a Warburg effect, which drives this full transformation that indicating that CIR6 evolved to be a major tumor suppressor, at least in the context of colon cancer. So when we published those results, we decided to follow two main questions. And I will briefly summarize the first one because we published it a few years ago to concentrate on unpublished data. And those two questions were, first, do other cancers care about CIR6? Is this an adaptation exclusively from colon cancer, or other tumors also where CIR6 is working as a tumor suppressor? And second, how early is this glycolytic switch? When is Warburg happening during the process of tumorigenesis? And this question has to do with what I told you, what the field believed was a late adaptation to support biomass. And I will come back to that in a minute. But first, are other cancers where CIR6 function also as a tumor suppressor? And the answer is yes. So this is just an example. If you glimpse at the graph from the Cancer Cell Line Encyclopedia, okay? You are looking here at cell lines from many different tumor types, and you look at loss or gains. Just as a general look, you can see a lot of blue, indicating that in many different tumors, CIR6 is rather deleted than amplified, suggesting that in many tumor types, CIR6 appears to be working as a tumor suppressor, and these tumors need to get rid of CIR6 to acquire adaptations. And here on the left, you see for pancreas, 60% of these lines exhibit a deletion of CIR6. And this is in the lines when we look at the TCGA at RNA levels, many of the pancreatic tumors exhibit downregulation of CIR6 at the level of expression. So we thought maybe in pancreatic cancer, CIR6 also evolved to be working as a tumor suppressor. And indeed, this is the case. I will just show you a couple of data slides because we published this a few years ago. What we found is a major role for CIR6 as a tumor suppressor in pancreatic cancer. If you cross, and we did this in collaboration with Nabil Bardisi, a friend and colleague of mine here at the MGH, who developed several of the genetic models of pancreatic cancer. If you cross it against the KRAS G12D mutation, okay, this 95% of pancreatic cancers carry a KRAS G12D mutation. But KRAS alone is rarely sufficient to give you full PDAC. Okay, only one animal in our cohort developed a PDAC, and very late. Most of the others never got full transformed pancreatic ductal adenocarcinoma. If you cross it to a CIR6 deletion, every single one of these animals develop aggressive disease. And more importantly, 50% in average of these animals develop metastatic disease with both liver and lung metastasis. If we do this in the context of both KRAS and P53, and these animals, all of them develop PDACs, even in this very aggressive model, getting rid of CIR6 pushed the curve, and some of the animals came down with disease as early as in 12, 13 weeks. And again, we see metastatic disease with liver and lung metastasis in almost 50% of these animals. And actually our model of the CIR6 deficient animals is one of the first mouse models of pancreatic cancer with lung mets. And we've been using this to compare metastasis to primary disease because the metastatic lesions come so fast that they don't have time to acquire new genetic drivers. And we think it represents a beautiful example of epigenetic and metabolic adaptations of metastatic cells. And we are using this system to understand what are the unique features of the epigenetic adaptations of metastatic cells. And at the end of the talk, if we have time, I can tell you about this project later. Interestingly, in the pancreatic cancer model, lack of CIR6 didn't give a metabolic advantage. Works by Alec Kilmelman and Ron DePino show nicely that the KRAS mutation drives a glycolytic switch, make these cells already carrying a Warburg adaptation. So why these tumors got rid of this chromatin factor? We needed to step back, look, and to make a long story short, we performed genome-wide ChIP-seq to look at changes in histone K9 and K56 acetylation, and we find one particular gene called LIN28B. This is called an oncofetal protein. It's expressed in mammalian cells very early during development and completely shut down in somatic cells. We found that it's shut down because of CIR6. It's a major repressor of LIN28B expression. And we found that pancreatic cancer cells get rid of CIR6 to allow upregulation of this oncofetal protein. And because LIN28B, and this is a MYC-dependent activation of LIN28B, we see LIN28B upregulated. It's inhibit the expression of a microRNA called LED7, and that allows the expression of several of its targets that are non-modulator of metastatic growth. Among them, IGF-2BP and HMGA2. Okay, so we found that lack of CIR6 drives the upregulation of LIN28B, and since this is a gene expressed early in development, what we were doing is de-differentiated these pancreatic tumors. And because they become de-differentiated, they were growing in such an aggressive fashion, much more aggressive in making metastatic disease. Now, we believe this represents a beautiful example of what is called an epigenetic plasticity. In the context of colon cancer, this colon cancer cells got rid of CIR6 to acquire a metabolic adaptation. In the context of pancreatic cancer, they didn't care about CIR6 for their metabolic features. They were already glycolytic, but getting rid of this chromatin factor, it's a chromatin factor, it regulates many genes. By getting rid of CIR6, they uniquely upregulated LIN28B, and this gave these pancreatic cells an advantage. So that for other cancer, and in data, I may have time at the end, I will show you that this is not true only for colon and pancreas. We have several other tumors where we see clear effects for CIR6 as a tumor suppressor, particularly in the context of squamous cell carcinoma that I will try to mention at the end of the talk. But for the second question now, so I show you in colon cancer, it's clear that CIR6 is regulating Warburg effect. How early is this Warburg effect happening during the process of transformation? And we decided to explore this question because, as I mentioned before, most of the people in the metabolism field believed that this was a late adaptation of rapidly proliferating cells. Rapidly proliferating cancer cells, they need biomass, so they sidetrack glucose to produce nucleotide in the pentose phosphate pathway, as I said, serine through one carbon metabolism, and lipids through acetyl-CoA. We have some data that suggested the contrary. We had some data that suggested that in certain cases, getting rid of CIR6 and acquiring expression of these glycolytic genes happen very early during the process of transformation. For instance, this is data from the NCIG60 database, where you have RNA levels in tumors, and you can see this is in colon, this is in pancreatic cancer. You have levels of CIR6 and the glycolytic genes in normal tissue and in carcinoma. And you can see very clear the decrease in CIR6 in most of the tumors and the concomitant increase in metabolic genes. And in colon, you can look at stages. And you can see that even at stage 1 colon cancer, and we have data from adenomas that show exactly the same pattern, already very early on, these cells are getting rid of CIR6 to increase expression of this glycolytic gene. This is not a late adaptation of aggressively proliferating cells. So we decided to explore this and say maybe transformed cells acquire this Warburg effect early during the process of transformation. And actually my postdoc at that time, Carlos Sebastian, he's now in Barcelona, he said, Raul, let's explore one step further. How do we know that these cells are acquiring an adaptation once they become transformed? What about normal cells that may be already doing this unique metabolic feature? What we know about normal cells that may be doing Warburg, and because they're carrying this metabolic feature, they may have already a head start. So I told him, look, Carlos, as far as I know, the only cells that are known to do Warburg are lymphocytes. All the pioneer work by Craig Thompson that actually brought Craig to do cancer metabolism was his work on normal lymphocytes that are clearly doing a glycolytic switch when they get activated. And some of this work that was also followed by Marsha with Arlene Sharp. So these are the only known normal cells that do Warburg. I told him, as far as I know, we don't know much about normal cells and Warburg. So we decided to explore in the intestine because we have the model and there is a unique feature of the intestine. So probably all of you know from the work of Hans Klever that the intestinal stem cells that were clearly described by Hans are carrying this unique LGR5 marker. They sit in the crypt and they have the ability to generate all the different population in the intestine. Hans published a beautiful paper in 2009 showing that if you eliminate the APC gene exclusively in the stem cells, in the intestinal stem cells, you get full-blown adenocarcinoma. If you do that from the transitional amplifying cells, you don't get tumors. So he said the intestinal stem cells are the precursor of what he called intestinal cancer stem cells. Okay, these are the population of cells that once transformed can get rise to full-blown adenocarcinoma. So we ask, let's explore what are the metabolic features of these cells before and after transformation. And we have the model, I told you, we generated this APC means CIR6 deficient animals that deleted CIR6 specifically in the intestine. And the first result we found was that if you don't have CIR6, we see an increase in the number of these intestinal stem cells. These are normal intestinal stem cells, not transformed. And we have a 25% to 30% increase in the number of these cells. We use a marker called Olfen4 that marks a lot of the stem cells in the intestine. And we see this clear increase. And if we give the animals dichloroacetate, the drug that inhibits glycolysis, we rescue the increase in the number of these cells, suggesting that the increase, this boost in the extra number of cells come from this boost in glycolysis. Now you may say this is not a major increase, but you can't bring that much more. You are filling the base of the crypt and still you need to maintain the Paneth cells in between the ISCs. This is as good as it gets for a normal crypt. Now, you can also make in vitro intestinal organoids. Again, we learned this thanks to Hans Klever. You can grow intestinal organoids that they resemble the structure, the anatomical structure of normal intestine. But what you may not know, that the number of organoids that you get when you purify crypts reflect the number of stem cells with which you started the culture. And again, when we look at the number of organoids, we got this 25, 30% increase in the number of organoids. What is more important, when we did this from animals that carry the APC min gene, and now you can grow organoids without R-sponding, which is the Wnt signaling factor, because they carry the APC min, they have constitutive Wnt signaling activated. You get transformed organoids. These are called spheroids. In the context of the transformed spheroids, then the lack of CIR6, the increase in glycolysis become much more robust. And you have a threefold increase in the number of organoids, suggesting that by pushing glycolysis, we were increasing the number of, in this case, intestinal cancer stem cells in the absence of CIR6. And in data I am not showing you here, if you treat in vitro these organoids with dichloroacetate, the organoids completely crash. So they need glycolysis to grow and proliferate. So I told Carlos at that stage, where we need to prove that these stem cells are more glycolytic. To my surprise, there were no clear reporters of glycolytic genes. We have fluorescent reporters for every gene in the genome, but no one made fluorescent reporters for metabolic genes. So we decided to do it ourselves. We put a 1.5 kb promoter of the PDK gene that I mentioned before, upstream of the M-cherry reporter. We made viruses, we infected the organoids in vitro. The organoids, as I said, it mimics the intestinal architecture, and the intestine has a lumen, the organoids have lumen and have intestinal stem cells. So you can basically look in vitro what are the particular cells that are doing glycolysis. And I told Carlos, don't expect a black and white picture. This is a metabolic gene. This is not a developmental marker or a stem cell marker that will be marking specific cells. Most likely we will see quantitative changes in these metabolic genes that hopefully will allow us to distinguish unique cells that are more glycolytic than others. And for me, this was a surprise. We got basically a binary type of experiment of staining. This is just autofluorescence in the lumen, but this is an organoid. These are the cells, the epithelial cells in the organoids. And you can see that only 4 cells in these organoids are lighting up with a reporter. Everything else is negative. So we have these very few rare cells that are expressing PDK1 and the rest are not. And the same is true for almost every one of the organoids we looked. Now I've been presenting this in few meetings, and I early on started to get the question, well, Raul, you are not really looking at endogenous glycolysis. This is a reporter with a 1 kb piece of PDK1. That could be an artifact that these are ectopically expressing PDK1. You are not really looking at endogenous glycolytic activity. So we spent almost a year trying to find a good antibody for the target of PDK1, which is 5-pyruvate dehydrogenase. And there is specific antibodies that detect the phosphorylated form of PDH. And we found a beautiful antibody that works in immunofluorescence. And when we did this in the organoids that carry the reporter, you can see these 2 cells here lighting beautifully up with the phospho-PDH. These are the only 2 cells that are marked by the reporter. So our reporter is a very nice reflect of endogenous glycolysis and can serve us to purify and characterize these cells. And again, in this picture, you can see that these are very rare cells that are doing glycolysis in a sea of negative cells. So we were actually surprised, and I told him, well, maybe really these are unique cells that are expressing these genes. They are doing glycolysis, and by doing glycolysis, once they get transformed, they have a head start. So I told him we need to prove that this has to be the LGR5 cells, these intestinal stem cells. Why they will be doing Warburg already if they are normal? One thing that I didn't tell you, different from other somatic cells, somatic stem cells that are mostly quiescent, like the stem cells in the hematopoietic system or like the stem cells in the breast, LGR5 cells are rapidly proliferating. And from data that was mostly coming from Hans Kleber's lab. So we said maybe because they are uniquely proliferating, they benefit from doing Warburg. So we said, okay, let's explore. We decided to stain for a marker of proliferation and our reporter. We said maybe we will see more cells proliferating in the absence of CEC6, and they will be proliferating more because they are highly glycolytic. And that's when the second surprise came to us. When we stained for Ki67, a marker of proliferation, and the PDK1 cherry reporter, we got a mutually exclusive pattern of staining. Here is in the organoids, and you see a lot of cells proliferating in green, and very few red cells marked with the PDK cherry, and none of the red cells are green. We took transformed APC organoids, we injected into mice, they are carrying the PDK cherry, you get tumors. And then we took the tumors out, and that we mark with the Ki67 and PDK cherry. Again, mutually exclusive pattern of staining, not a single red cell was marked with the marker of proliferation. So this for us was a surprise. We have here a population of cells that are not dividing, that are clearly glycolytic. What are these cells? in both the context of normal intestine and in transform. So we have to step back. We went to the literature, and there is in the intestine a population of cells that were very nicely described by several groups, including particularly Ramesh Shivdasani in Dana Farber and Calvin Kuo in California. These are called the +4 stem cells. They have several features. They are not real stem cells. These are actually enteroendocrine secretory cells in the intestine, quiescent. But if you eliminate the intestinal stem cells, the LGR5, they have the capability to de-differentiate, become LGR5, and drive regeneration of the intestine. So we decided to use our phospho-PDH antibody to look which cells were particularly doing glycolysis in the normal intestine. And when we did that with the phospho-PDH antibody, you can see in every one of these crypts a single cell lighting up like crazy with the antibody. The transitional amplifying cells show some glycolytic staining. Some of these cells are doing glycolysis, but it's mostly this black pitch black cell in the base of the crypt that is staining with the phospho-PDH antibody. And it's the same result if we look by immunofluorescence, and since the antibody is working by immunofluorescence, we decided to take the LGR5 GFP mouse from Hans Kleber and stain for the phospho-PDH. And you can clearly see in this example here, that these cells are not the LGR5. The LGR5 are the green cells on the base, and it's one cell above that is staining with the phospho-PDH. As I mentioned, these are enteroendocrine cells, and many of these cells stain positive for a marker of enteroendocrine cells called chromogramin A. So we co-stain for chromogramin A and phospho-PDH, and here you have 3 cells that are clearly lighting up, and actually 95% of the chromogramin A cells in the base of the crypts were also phospho-PDH positive. So we believe we identified a unique metabolic feature, these highly glycolytic cells, in these quiescent +4 cells that appears to give these cells an advantage in the context of a normal intestine, but once they are transformed, coming with this unique feature gives these cells an advantage. Now, several laboratories, including those of Ramesh and Calvin Kuo, shows that the ability of these cells to de-differentiate and become LGR5 is critical for the ability of the cells to regenerate following damage. And one way you can induce damage is by irradiating the mice. The same irradiation you give to patients undergoing radiotherapy or chemotherapy, the main first symptom that these patients acquire is diarrhea. And this is because you wipe the crypts in this intestine, and they need to regenerate. So here, if you stain for Ki67 6 days after the irradiation, you see very nicely the regeneration of the intestine. If you do that in the context of the drug that inhibits glycolysis, dichloroacetate, you see a major inhibition in the ability of the cells to regenerate, indicating that the glycolytic phenotype of this +4 is critical for the function, for the regenerative capacity of these normally quiescent cells. And you don't need to see much on this slide. This is human intestine, and all I want you to see, these are 3 dots that are red, phospho-PDH positive, the same 3 dots are green, chromogramin A positive. And so just to show you that in the human intestine, we see the same rare population of phospho-PDH chromogramin A positive cell. So our results is not an artifact of a weird artifact of mouse cells, and these cells are present also in the human intestine. So what I show you today is that we identify a unique metabolic feature. This Warburg effect doesn't happen only in transformed cells and lymphocytes, and we have these unique rare +4 cells that are normally quiescent, that are highly glycolytic, and the increasing glycolysis in the absence of C6 appears to drive a push to increase the number of the LGR5 stem cells here. Carrying these metabolic adaptations, once these cells acquire a mutation, we believe it may be giving these cells a metabolic head start, and that's why they end up with more cancer. Now, one obvious question is why these normally quiescent cells are doing work. I kept telling you again and again that this is an adaptation for biomass of rapidly proliferating cells. Why quiescent plus 4 cells are so highly glycolytic. So what I can tell you is that we found that this is work from Ramesh, showing that when you compare the transcriptome of BMI1, these are the plus 4 cells, with the LGR5 stem cells, the BMI1, the plus 4 cells have a much stronger antioxidant signature of genes. And the same is true if you measure reactive oxygen species. We took an organoid, this is a staining for cell rocks and phospho-PDH, and you can see one cell here that is stained for phospho-PDH has much less levels of cell rocks compared to the neighboring cells. And this is quantified here. Moreover, if we treated organoids with an antioxidant drug, this is N-acetylcysteine, we get much more organoids. And remember, I told you, the number of organoids reflect the number of stem cells with which you started this culture, suggesting that these plus 4 cells may have adapted to do Warburg to reduce respiration in the mitochondria, to reduce the production of reactive oxygen species. And there is a functional consequence of the ability of the cells to do glycolysis. And this was a request by one of the reviewers. The paper is under revision now. We purify the M-cherry positive with our reporter, the M-cherry positive, M-cherry negative. We injected this into mice, and only the M-cherry high positive cells were able to give us tumors. None of the conditions we tried with the M-cherry low cells were able to give us tumors when injected into mice. So I have 5 more minutes. I was asked to finish at 12:50, so we have time to go through questions. I will just mention briefly a parallel story that I told you at the beginning. When we were trying to understand, is this an adaptation? Which tumors get rid of CIR6 to acquire a metabolic advantage? We started in parallel a project in squamous cell carcinoma because this is a tumor that we know very little about metabolic features. And second, this is one of the few tumors where we have very well defined, at least in mouse models, what are called the tumor propagating cells. Some people call them cancer stem cells, others call it tumor propagating cells or TPCs, and there are specific markers for these cells that has been described. So we decided to investigate whether squamous cell carcinoma also may benefit from getting rid of CIR6 and whether they benefit by acquiring a metabolic advantage. And long story short, as I said, I don't have much time, so I will go rapidly through the results just to show you that similar to in the context of colon cancer, absence of CIR6 increased glycolysis, but different from colon, we see the glycolytic shift happening in these basal rapidly proliferating cells. So you can see here expression of GLUT1 exclusively in the basal cells of these dysplastic cells. And these are tumors that we derive in the skin by treating this animal with a protocol called the MBA-TPA. These are oncogenic drugs, and that treatment with these drugs cause these animals to develop papillomas. And in the absence of CIR6, we saw an increase in the number of the papillomas that you can see here. And actually, some of these animals develop full squamous cell carcinomas. We have an increase in the size, increase in the number. And you can see nicely here that GLUT1 is highly expressed specifically in the basal rapidly proliferating cells. More importantly, when we stain for markers of these tumor propagating cells, alpha-6 integrin and CD34, we see that in the absence of CIR6, a 3-fold increase in the number of these stem cells, which is here. And if you treat this animal with a glycolytic inhibitor, DCA, most of these cells goes away. Why they adapt to do that? Again, the same as we saw for colon, we see a push in glycolysis, a push in stem cell markers. Okay, these tumor propagating cells express specific stemness markers that are upregulated in the absence of CIR6, and a clear increase in the number of metabolic genes. Why they do this switch? We were able to perform metabolomics in this case by getting rid of CIR6 in a couple of squamous cell carcinoma lines, and we saw a clear, we don't need to go, I'm sorry if I'm going fast, you don't need to read this, just to show you a clear shift that explains a boost in what is called the oxidative branch of the pentose phosphate pathway, that is in charge of increasing reduced glutathione against oxidative stress. And here you can see the increase in reduced glutathione by metabolomics. Again, suggesting that the reason these cells do the switch to warburg is to sustain antioxidant defenses. And just one last piece of data, one of the reviewers said, all the metabolomics data you show is in vitro in cancer cell lines. You need to convince me that the CD34 alpha-6 integrin in the normal, in the tumors in vivo are doing glycolysis, are increasing glutathione, reduced glutathione. This was quite a difficult question to ask, because as you know, to perform metabolomics, you need a minimum number of cells, and this CD34 alpha-6 integrin are an extremely rare population, but we were lucky to collaborate with Natalie Agar in Harvard. She's at the Brigham Hospital, and she has the capability to do what is called MALDI-MSI, mass spectrometry imaging, which is basically, it's like a microscope, but instead of a microscope, it's actually a mass spec that can pixel by pixel acquire information, metabolic information on a slide of a tissue. And when we did that, these are some examples of our tumors. You have here normal skin and 2 tumors, and you have different metabolites. Here is glutathione, and we are capable of taking one slide, staining with CD34 and GLUT1, and a neighboring slide analyzed by metabolomics. And this is glutathione, an image for reduced glutathione. And you can then ask the computer, do the areas that are staining positive for CD34 and GLUT1 exhibit more or less reduced glutathione? And this is quantified here, and you can clearly see a very nice increase in reduced glutathione in the CD34 GLUT1 compared to the more differentiated keratin 10 positive CD34 negative cells. And this antioxidant protection is functionally relevant. If you treat these cells with N-acetylcysteine, we see an increase in the number of these stem cells. So I will stop there. I don't have time to show you. We also did single cell RNA-seq on these stem cells, and we identify a unique subpopulation of the stem cells that were clearly glycolytic, indicating that these cancers are exhibiting what is called a metabolic heterogeneity. That I don't have time to show, but suggesting that the Warburg effect, we need to rethink what we call the Warburg effect in cancer. We believe that the field needs to re-evaluate. It's really an adaptation of rapidly late proliferating cancer cells, what we know about normal cells in their ability to perform aerobic glycolysis. And second, when we are looking at tumors, is this an adaptation that happens in the whole tumors, or are we talking about few specific cells within a tumors that have the capability to do Warburg? Is this an heterogeneous feature of cancer cells? So this is basically what I wanted to leave you with today. And I will acknowledge, these are the people, I've been acknowledging them throughout the talk. These are the current people that I have in the lab, with a lot of former members, many amazing collaborators. And this is a current picture. Again, I was mentioning some of the people that were giving and that were making this experiment throughout the talk. And with this, I will stop and take questions. So you have a few minutes for those.
That was just terrific, Raul. A lot of really exciting data. So there are several questions here. And so I'll start with the first one from Quang Ting Kuo. He says, great talk. Since pancreatic cancer is a highly heterogenic cancer, I'm wondering if the SIRT6 downregulation is in cancer with a specific type of mutated KRAS, or is it generally downregulated in PDAC regardless of the status of KRAS mutation?
That's a great question. And when we look at the TCGA, we see SIR6 downregulating in most of the tumors that we look. Again, 95% of these tumors exhibit KRAS mutation. And I know that Bill Hunt, particularly, has been working specifically with the KRAS wild-type PDACs in recent years. We didn't look at this particular subgroup, But what I can say from just looking in the TCGA, most of the tumors that we look for CIRS6 level appears to downregulate CIRS6. We didn't look specifically on those 5% that don't carry KRAS mutation. We should do that. But definitely appears that most of the tumors we look downregulate CIRS6 in the context of PDAC.
So the next question is regarding HIF-1 alpha. Since HIF-1 alpha is post-translationally stabilized by the depletion of alpha-ketoglutarate, an intermediate of the TCA cycle under aerobic glycolysis, do you think that the transcriptional control of HIF by SIRT6 and post-translational control by alpha-ketoglutarate go hand in hand?
That's a fantastic question, and I will need to clarify. I didn't say that HIF-1 alpha was stabilized at the level of transcription. And actually, we don't see changes in RNA level for HIF-1 alpha. It is stabilized post-translationally. And what we believe, and at that time when we published that paper, this was a question from a reviewer, how is HIF-1 alpha stabilized? The only thing I can say, there were a few papers showing that lactate itself has the ability by mechanisms that were not known then, and it could be related to the accumulation of alpha-ketoglutarate. Lactate itself appears to play a role in stabilizing HIF-1 alpha. And we did some experiment in vitro to confirm that. We didn't look at alpha-KG itself, and we probably have now tools to do that. You could probably treat cells with IDH mutations, so you reduce alpha-KG. We haven't done that, and so I can't tell you 100% how is a HIF stabilized, but it's also in the context of CIRS6 depletion happens post-translationally, not at the level of transcription.
Your next question is by William Beck. I think he's referring to the colon cancer work. He writes, how can such a small population of cells drive a tumor? What transforms them? How do they adapt to glycolysis?
Yeah, thanks, William, for the question. I wish I knew. So if you look at the staining, I show you the staining of the organoids and the tumors stained with the PDK cherry Ki67. There were much more cells in the tumor that were staining positive for PDK cherry. So I believe that in the context of the normal intestine, these are a very rare population. Once they are transformed, I think many more cells start to express and become glycolytic. It's interesting that still none of them are expressing proliferation markers. It's in the context of the tumors, at least in the colon cancer, still are the non-dividing cells that are the ones that are glycolytic. They are much more, so these populations are clearly much more than what we tend to believe for a rapidly proliferating tumor, but there are works by many laboratories showing when you look at quiescent cells within a tumor, they are actually quite much more than people predicted before. It is a mixed population, and I believe a lot of them are glycolytic. How can you explain the number of the cells and how a tumor keep growing on cells that are quiescent and glycolytic? Why they switch? Why they stop being, at least in the context of colon, why they stop doing glycolysis? If this is an advantage for proliferating cells, I don't know, and we are right now starting to explore that. And for this, of course, we need genetic models, and Carlos, my former postdoc, is making this reporter mouse to look on normal cells and other cancer models.
So I want to ask this next question. It shifts gears a little bit and asks about therapy. Do you think it's a good idea to use SIRT6 inhibitors for cancer or any other disease treatments?
Well, SIRT6 inhibitor, I would say no, because you will get more tumors. This is a tumor suppressor, so if anything, you will need to activate SIRT6 to push, to inhibit the Warburg effect or LIN28 in the context of pancreatic cancer. And as you know, it is much, much more difficult to activate this enzyme than to inhibit them. So if you ask me, I would say yes, I think that activating SIRT6 will be fantastic if we can do it. I wish we could do it, and I know there are actually a couple of companies trying, but it is much more difficult to activate this enzyme than to inhibit them. So we will see what the future of the part, but if not, if you can't go for the enzyme itself, it could be another strategy to inactivate or downregulate some of the targets. Usually when you get a chromatin factor changing is because most of the targets that go up work as a group. So targeting a specific one may not do it, but it's a question of trying, I guess.
I want to add, end with a question by Bill Kalin. He says, by the way, even a stage 1 tumor will contain hypoxic cells, which will upregulate HIF and glycolysis. Does that complicate your results? How do you consider that?
Thanks, Bill. Yeah, I don't know. I don't think it's complicated the result because I didn't say, we did use in a couple of our models markers of hypoxia, and we didn't see much hypoxia. We didn't do it for the intestinal ones. We did it in another model where we were looking at the glutamine deprivation and in the squamous cell carcinoma cells. We didn't see, when we use the marker for hypoxia, that these cells are experiencing hypoxia. I can't tell for the +4. I think Ramesh have some data showing that these cells are not hypoxic, but you mentioned early stage 1 colon carcinoma. We didn't look at these specific cells, so I can't tell you whether the cells, once they become transformed stage 1 early, they may be experiencing hypoxia, and this is pushing this glycolytic switch also. That will be something to look. It may complicate if we are trying to argue that this is exclusively aerobic glycolysis. In that regard, yes, we will need to be more cautious and say that maybe some of the cells are experiencing hypoxia, but we didn't measure specifically at early stage 1 transform.
So thank you again, and thank you everybody for your questions. There are a number of questions we didn't get to answer due to time's sake, and Raul will have a chance to type his answers. Thank you all.